Okay, so let's start. In this video, we will see the principles and primer of PCR, means on what principle the PCR works, and how the primer is designed, and what are the qualities that a primer should have. So let's start our video. So first of all, polymerase chain reaction, that is PCR, is used to amplify small and targeted segment of DNA to produce millions of copies of specific gene segment. It is developed by Kerry Mullis in 1993. What are the requirements? That is thermal cycler, it is PCR machine, thermal cycler, DNA template, forward and reverse primer, tag polymerase buffer, deoxynucleotide triphosphate, water and nucleotide. These are the requirements. Let's understand this in whiteboard. Okay, suppose this is a DNA segment. This is a double-stranded DNA segment. It is 3 prime, 5 prime, 5 prime, 3 prime. Okay, we need to amplify this DNA. So first of all, what we will do, we will separate both this strand from each other. First step, we need to separate this strand from each other. For that, we will increase the temperature up to 95 degrees centigrade. When we increase the temperature up to 95 degrees centigrade, then both these strands get separated. Okay. This step is known as denaturation. Both these strands get separated. This step is known as denaturation. And now each separated strand will act as a template. So this will act as a template, and this will also act as a template for synthesis of new DNA strand by PCR. Okay. So after denaturation step, the next step is, denaturation is the first step. Second step is annealing of primer. Annealing of primer is the second step. Here primer is attached. Here the primer is attached to each template. This step is known as annealing of primer. Okay. And after annealing of primer, the last and the third step is extension. Okay, so we will see this. Okay, extension step. This is third step. Here primer is attached. In the second step of the PCR, that is annealing of the primer. In third step, extension of primer, what will happen? Using the 3' OH of the primer, DNA polymerase will extend it in this direction. Here, using the 3' OH, DNA polymerase will extend or synthesize the new strand depending on this template strand. This strand will synthesize depending on this template strand. Here one thing is important to notice, that in denaturation step, when temperature is increased to 94 or 95 degrees centigrade, most of the DNA polymerase is degraded. So here we use a special type of DNA polymerase, that is tag polymerase. Okay, we use a special type of polymerase, that is tag polymerase. This tag polymerase is stable in 94 to 95 degrees centigrade. So we use this polymerase. Otherwise, if we use other polymerase, then those polymerase will get degraded or denatured in 95 or 94 degree centigrade. So PCR occurs in 3 steps. First step is denaturation, where both the strand will get separated. by increasing temperature to 95 degrees centigrade, and each strand will act as a template for synthesis of the new strand by Taq polymerase. Second step is annealing of the primer, and third step is extension. Okay. Then two types of primer is there. One is forward primer and one is reverse primer. So what are these forward and reverse primer? Suppose we get both this strand by separating or by denaturation steps. So it is 3 prime, 5 prime, this 5 prime, this 3 prime. Here the primer will attach. Okay, primer will attach. It will be 5 prime and it will be 3 prime. Okay, here another primer will attach. It will be 5 prime end of the primer. It will be 3 prime end of the primer. Okay, so it will synthesize in this way and it will synthesize in this way. Okay, extension will take place in this way. Extension from this primer will take place in this way. So this primer will be called as forward primer, and this primer will be called as reverse primer. Okay, means left to right is forward primer. Okay, and right to left is reverse primer. Okay, forward and reverse primer. Then nucleotide, we need nucleotide also. How? Suppose these are separated strand by the process of denaturation. Primer is annealed. Here it is 3 prime OH and here it is 3 prime OH. Now to extend from this primer we need to add nucleotide. From where this nucleotide will come? So in the PCR mixer, we need to add nucleotide also. So that this nucleotide can come and bind to this 3 prime OH depending on this template, and it can extend the new strand. Okay, so the new strand will get extended in this way. Okay, nucleotide will come. Okay, it will extend in this way. Now what nucleotide will bind to this? What nucleotide? Adenine, guanine, cytosine. What nucleotide will bind here? It will depend upon this template. Okay, what nucleotide gets bind here will depend upon this template, but all the nucleotide we have provided in the PCR mixer. Okay, what will be the other component of the PCR mixer? Taq polymerase. We need to add Taq polymerase. Okay, we need to add a buffer. Also. Okay, we need to add water also in this PCR mixer. Okay, so see here, buffer, Taq polymerase, because it is stable at 94 degree to 95 degree centigrade is stable. Forward and reverse primer, DNA template, and thermocycler is the machine. Nucleotide we have to add. Otherwise, if we do not add any nucleotide, then how the new strand will synthesize? Okay, how the new strand will synthesize? For the synthesis of new strand, there must be nucleotide that can be added to the 3 prime OH of the primer. Okay, so next, target DNA. Target DNA is the DNA to be amplified. Segment represent a small part of large and complex mixture of specific DNA of a genome. Primer limit the DNA sequence to be amplified. So let's understand this in whiteboard. Suppose this is a DNA. Okay, this is a DNA, and here A gene, B gene, C gene is there. Okay, or you can say segment A, segment B, segment C, or gene A, B, C is there. We need to only amplify the C gene. We need to amplify only C gene. So what we will do, we will synthesize some primer. We will synthesize some primer that specifically bind to this region and specifically bind to this region. Okay. For that, we have to know the sequence of this region. Without knowing the sequence of this region, we cannot synthesize a primer that specifically bind to this region. So, first of all, we need to know the sequence of this region. Then, primer will be synthesized that will be complementary to this. That's why this primer will bind in this region. Okay, primer will bind here. Okay, it is specific primer. After that, during extension, it will synthesize to this portion. Okay. Here, B segment is not amplified, A segment is not amplified. Only C segment is amplified. Why? Because we use specific primer, okay, that bind to the flanking region of the C segment. Okay. Suppose we here A is in B segment and C. Suppose our primer binds here. Okay, our primer binds here. Okay, our primer binds here. Now extension will cover A segment, B segment with C segment. This will amplify all these segments. It will amplify this segment, it will amplify this segment and C segment also. But we do not require the B and C. That's why we will use the specific primer that binds to the flanking region of C. Okay? Then primer design. Primer should not be very short or long. Self-complementary sequences should not be greater than 3 base pair, and melting temperature of the two primers should not differ by greater than 5 degrees. EC content should be 40 to 60%. Okay? Should not be too small or too long. Let's understand one by one criteria per DNA primer. Okay? Suppose should not be too short, or long. Okay, suppose there is a DNA molecule, of 10 million base pair. Okay, and we designed a primer of 8 nucleotide. Then what is the probability of finding attachment site of this primer in this DNA molecule? What is the probability of finding the attachment site of the primer in the DNA molecule of 10 million base pair? So we know there are 4 nucleotide bases and our primer is 8 nucleotide. So it will be 536 base pair. That means in every 65,536 base pair there will be an attachment site of this primer. Okay, means in every 65,000. Suppose here is the DNA, total DNA. From here, suppose one attachment site. From here, one attachment site of the primer. Starting from this, after 65,536 base pair, then there will be another attachment site. From here, again after 65,536 base pair, from here another attachment site will be there. Okay. From here again 65,536, after this much base pair, another attachment site of the primer will be there. Okay. So in 10 million base pair there will be more than one attachment site of the primer. Okay. But if we synthesize primer of 15 nucleotide, then 10 to the power 50, okay, it will be around 107 crore base pair. Okay, means suppose this is a DNA of 10 million base pair. So suppose here is one attachment site of the primer. From this attachment site, after 107 crore base pair, there will be another attachment site of the primer. Okay, so starting from here, after 107 base pair, it will be somewhat here. Okay, here, but our DNA is 10 million base pair. So in this DNA there will be only one attachment site. Okay, if my DNA length was like this, then there will be two DNA binding site, but my DNA is up to this mass, 10 million base pair, so there will only be one attachment site of the primer, because the next attachment site will occur after 107 crore base pair, but this much base pair is not present in our DNA. Okay, that's why primer should not be too small or too short and too long. An ideal primer should be 18 to 20 base pair long. It should be 18 to 20 base pair long. Okay, next is self-complementary sequences should not be greater than 3 base pair. Let's see this in whiteboard. Suppose we have designed a primer like AAA, TTT, CGAT. Okay, we have designed this primer. Now you see A is complementary to T, A is complementary to T, A is complementary to T. So there is possibility that this 3A bind with this T, CGAT. Okay, this A bind with this T within the primer itself. Okay, if such cases happen, then how this can attach to our DNA sequence? Primer binds upon itself. Within the primer there occurs binding. Okay, self-complementary. This is self-complementary. Within the primer they bind to each other. So it is not possible that it will bind to DNA. Okay, so okay, A, T, C, G. Okay, it is okay, because self-complementary is less than 3. There are 2 complementary bases, it is not 3. But here you can see the complementary is 3. Complementary bases present is 3. So it will not work as primer. Okay, because if we use this as a primer, there is high chances that self-complementarity or self-ligation will be there within the primer itself. So if self-complementary is there, then it will not bind to the DNA, because the primer will bind within itself. Okay, second requirement. The third is the melting temperature of the two primers should not be differed by greater than 5 degrees centigrade. So let's see this also. We have discussed that forward and reverse primer. Suppose one primer is there, forward and reverse primer. The melting temperature of this suppose is 50. Okay, and the melting temperature of this must be 45 or 55. Must be 45 or 55. It cannot be 40 or 60, because if it becomes 40, the difference with this primer will be more than 5, and it becomes 60, the difference with this primer will be more than 5. It cannot happen, or it is not acceptable. Okay, then GC content should be 40 to 60%. Okay, GC content within the primer should be 40 to 60%. Suppose I have designed a primer A8 G C C Z T C Z C C C T. Okay, suppose I have designed this primer. So how many nucleotides are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So 13. Okay, 13, 14, 15, 16, 17, 18, 19, 20. Okay, 20. Total number of nucleotide is 20. Okay, How many G and C are there? 1, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 nucleotide. What will be the percentage? Okay, so 60%. Okay, so this will be, this primer qualifies, this primer qualified for 40 to 60% criteria. 40 to 60% criteria of GC content, it qualified. Okay, it has 60%. Okay, so it is a good primer. Next, probability of a complementary sequence of a primer to be present in DNA is calculated by this formula. That means the probability of complementary sequence present in DNA is calculated by K is equal to G by 2 G plus C into 1 minus Z by 2 whole A plus T. Okay, G implies the GC, relative GC content. Relative GC contents mean total number of nucleotide up by Z plus C. This is relative GC content. Okay, relative GC contents. Remember this term, G means relative GC content. Okay, and this is Z plus C. Simply Z plus C is not relative content. Okay, Z plus C is different and relative Z plus C is different. Okay, then degenerate primer. When partial sequence is available, not the complete sequence, mixture of primer, all of similar sequences but variation at one or more points, generally used in protein sequences. So let's see what is degenerate primer. Suppose this is a DNA sequence, we need to amplify this portion. We need to amplify this portion. For that, we need to synthesize a primer that specifically bind to this region. Okay, we need to synthesize a primer that specifically bind to this region. For that, we have to know the sequence of this region. We have to know the sequence of this region. Because if we know the sequence of this region, then depending on that sequence, we can synthesize primer. Okay, but if we do not know the complete sequence of this region, if we do not know the complete sequence of this region, then we synthesize degenerate primer. Then we synthesize degenerate primer. In this degenerate primer, there are mixtures of primer. These degenerate primer have mixtures of primer. Okay, mixtures of primer means, suppose one primer is AAAA, AAGCTC. Okay, another will be ATGCTC, another will be AAGCTT, another will be like AGGCTT. Okay, these are different primer, and we mix this primer in a tube to form a degenerate primer. Degenerate primer, because we know some of the sequences, some of the nucleotides here, but we do not know the complete sequence. That's why different primers are mixed to form a degenerate primer. Okay, so PCR steps: first, denaturation, heating up to 94 to 96 degree for 10 to 20 minutes, causing the complementary sequence to separate. Okay, tech polymerase is used. We have seen this. Then annealing, temperature is 2 degree lower than denaturing temperature, primer attached to single strand DNA. Okay, we have already seen this. And temperature is lower than 2 degree lower than annealing temperature. Okay, denaturing temperature. Then elongation is done at 72 degrees centigrade. Please remember the temperature. 72 degrees centigrade is elongation done. Tech polymerase starts filling the missing nucleotide in 5' to 3' direction. Okay. Then amplified product is called amplicon, and this is the number of product we can find by n naught into 2 to the power n. Number of amplicons we can find by this formula, number of amplicons. Okay, amplicons. Okay, number of amplicons can be found by this formula. Suppose a question comes like 100 cells are there and 10 cycles of PCR is run. 10 cycles of PCR is run. So what will be the number of amplicons? So number of amplicons will be n will be 100 into 2 to the power 10. This is n naught into 2 to the power n. N is 10. Okay. So the answer will be this. So this was all about the principle and designing a primer, what qualities primer should have. If you find our video, if you like our video, please share it, subscribe and share among your friends. Thank you.